I'm going to ask Nina, of course, to lead this. When targeting CD38 fails a patient, where do we go? And obviously BCMA has really been the talk of the meeting. So Nina, would you like to lead this, please, and talk to us a little bit about what caught your eye for BCMA-targeted approaches?
Yeah, so BCMA is a great target for cellular therapy for myeloma, and there are 3 modalities to do that. One is the CAR-T, for which the CARTITUDE data was presented. That is the U.S. adaptation of the LEGEND data. And so the CARTITUDE data, the first cohort was presented 100% response rate. So that made us feel good because we were worried about the only 3 prior lines in the Chinese data. And actually, concomitant with that, the LEGEND 2 data had further follow-up, and the PFS had improved from 15 to 20 months, which made us think maybe we should be using it in earlier lines. So that kind of went with that data. There was an early press release for the KarMMa-1 phase 2, in that the overall response rate for all cohorts is around 73, I believe, %, but actually for the 450 million cell dose is higher in the 80s, and the PFS is 11.3 or 4 months for that dose. Pretty much mirrors what I was expecting because they're going to probably go with the 450 dose, and these patients were sicker than the original Bluebird 401 trial, which was published in the New England Journal, meaning these patients were actively progressing when they got on the trial. And then there were a few other interesting CAR-T abstracts. Basically, I don't think anything is going to make a difference until we have PFS data. And for some of these, like the BB212 and 7, where the premise is you want to have the cells live longer, really duration of response. If you respond, do you respond for longer? So that data is pending. The other modality we have is BCMA bispecific T-cell engagers. This was my pick abstract for the entire meeting as far as BCMA goes, because it's a patient-friendly weekly and then every other week and then monthly schedule for a BCMA BiTE or bispecific engager, and they had 90% response rate in their active dose level. So that was really impressive. Question is, in myeloma, do you give drugs forever? I don't know. A lot of interesting infections. We'll see. And then finally, there is the BCMA-ADC, which I thought we were going to see, but we don't have it at this meeting. I think we'll probably know more later. The DREAM-1 study had originally showed a 60% or so response rate, 12-month median PFS. Some issues with the corneal toxicity. I go back and forth about this because I think that ultimately this is going to be manageable if you're that far into treatment, and if somebody wants a community-friendly treatment that's effective, I think we'll figure out how to manage it. There's been talk of that being a little bit more elucidated in the DREAM-2 data that'll be published. So those are the things that really are looking at BCMA. I think we need to use it earlier. And just one shout out to the BiTE AMG 701 program. Since 420 has been dropped due to continuous infusion, there was a preclinical data showing that lenalidomide may enhance that efficacy. And I think it's important because we're talking about BiTEs. The reality is it's not going to be single agent in the last line forever. It'll probably move up. So
yeah, and I must say I was also incredibly impressed by the BiTE data led obviously by Dr. Costa and that very exciting platform. That to me was quite remarkable. Sagar, just to build on that, and obviously that is the Celgene BiTE, exactly the 269. So if I may, Sagar, obviously both you and I have been very involved in the belantamab mafodotin story, GSK 916 or Belamaf for short. Thoughts on this? Because obviously we didn't have our late breaker, but that was for reasons simply of selection, I suspect, up against a phase 3, totally understandable. We'll be obviously making that information more public very soon. Just additional things that we can share.
Yeah, I mean, I think it's important, Nina, you mentioned the DREAM-1 study and the prolonged PFS and response rate in that. If you look at the DARA-resistant subset of DREAM-1, the response rate was actually in the mid-30s, and the PFS was 3 to 4 months. So I think we have to be, because DREAM-2 is a pretty resistant group of patients.
And you had to be DARA refractory.
Yeah. And they try to mirror it to the CAR-T and all that.
Correct. So I mean, I think if you're comparing apples to apples to apples, PFS may be different than what we saw in the DREAM-1 study.
And I would expect that.
Yeah.
And I would make that point then in terms of Luciana's data with the bispecific T-cell engager. I mean, the good part, it's a top 88% with DARA refractory.
Oh,
but we have no idea about durability of response.
We don't know.
Not yet. I agree. But certainly, what I loved was the convenience of it. I mean, it's a weekly infusion. and it doesn't.
And then it's eventually monthly.
Exactly.
Yeah.
So I think there's a practical application to it, which could be really game changing. I would echo exactly what Sagar's said. I think the important point about Dream 2 is that it embraces this truly penta-refractory population, and we were very, very pleased with how it performed. I would say, in terms of the corneal toxicity, Nina, we really do understand that better. It's not corneal ulceration, and it is not steroid responsive.
Right. And it's not necessarily symptomatic. It's graded based on
And I think what we found is it's very manageable. And if you reduce dose, and fascinating, the expanded access program, they've been dividing doses, literally the split dosing has become a thing. I'd love other comments from the panel on the BCMA space because it's so exciting. So if I may, Ken, and then I'll ask Amrita and Ajay to comment.
I guess the points I would make are, one is that obviously it's a very exciting. I'm gonna make 3 points I guess instead. One is very exciting, obviously, but two is CAR T and now BiTE. I mean, the BiTE data I think was powerful. It tells us we all thought the immune system's gone by this point in time, but now you're engaging a T cell and now it's doing a fantastic job. So maybe we're not so far off in terms of what we can do with the immune system late in therapy. So the data's outstanding, but it also teaches us a bit about the biology of myeloma late on, maybe educating us, at least changing our minds. I think the ADC I think is a fantastic way to go because we're thinking about CAR T, BiTEs. These are things that I still think at this point in time, these are all academic center, all specialty center kind of drugs, and the community really needs something else for these individuals who've failed lots of things. And BCMA-targeted therapy with an ADC is a fantastic way to go. And again, hopefully we can work on how to help them figure out how to get an ophthalmologist or an optometrist involved to help grade these things and what's the right thing to do, holding, split dosing, ways to figure things out will be really important for getting it truly into the community where they really need it.
It's fantastic. Amrita, other thoughts?
I think the thing that excited me a lot about the BCMA bispecific data was the idea that now we have bispecifics in myeloma, and we have, all of us are on many conference calls at strange hours for several other targets. And so I think this is just the beginning of the story. We're going to hear the GPRC5 story in terms of CAR T as well as in bispecifics, FCFR5. So I think this is just tremendous as a whole. As Sagar said, I forget it was the platforms or the buckets. I think we're going to have some new buckets in the future for patients.